So in chapter 6, we're going to look at a different aspect that we haven't discussed, and that is energy transfer and energy transformation. All of the energy transfers and transformations that happen within cells and between organisms and within ecosystems, no matter what level on the hierarchy that you're focusing on, there is energy that's being transferred and transformed. And the type of energy that the source of all energy on Earth is really the light from the sun, so sunlight. The heat is not very useful. If we could figure out a way to use the heat, especially in Texas in the middle of the summer, that would be a huge breakthrough in technology. But right now we typically look at the light from the sun as the important source, the important energy that comes into the biosphere. And the light energy is transferred and transformed by organisms we call producers, mostly plants, although there are some other organisms that are producers. But they transfer and transform that light energy into chemical energy. And by that we typically mean molecules, biomolecules. So the light energy is converted into chemical energy by the producers. And so think about glucose and other molecules and macromolecules. So the energy from the light is transferred and transformed by the producers into chemical energy. The other organisms, consumers, are mostly how we classify most animals, although some animals are decomposers like worms. Those are animal decomposers. But you have consumers and decomposers that use that chemical energy, and they use it for lots of different things, but ultimately everything dies, and ultimately all the energy eventually gets transferred and transformed into heat. And you'll remember that heat is a wasted form of energy. So really the flow of energy through all of life always goes from the form of light into some kind of chemical form, and it might be transferred and transformed into many chemical forms, but eventually it will dissipate as heat, right? So that is that flow of energy through the system, or what you could call bioenergetics. So let's talk a little bit about metabolism, that word. The definition of metabolism, it's the totality of an organism's chemical reactions. Or if we were talking about metabolism for a single cell, it would be the totality of the cell's chemical reactions. So you can look at it on a whole organism scale. You can even look at metabolism on a larger than an organism. You can look at it in terms of the whole population or the whole ecosystem. And so the metabolism is the totality of the chemical reactions in that system that you're looking at. And like we said, it's all about energy transfers and transformations. And we'll talk about what that means exactly. All right. So you have the matter of the cell, or the organism, which is the atoms and the molecules. We've learned about those. And now we're learning about the energy. All right. So both of those pieces are important for studying organisms, cells, ecosystems. Those are the two important pieces. So there are things within cells, and actually some of these can happen outside of cells, but typically we think of them happening inside of cells. A metabolic pathway. Metabolic pathway is just a series of transfers and transformations that occur in a certain sequence, almost like an assembly line. This little circle gets converted into a little triangle, and then that triangle gets converted into a square, and that gets converted into a oval, and then it gets converted. And this is just a simplistic example, but it's these step-by-step conversions. The whole series of metabolic changes that occur to this yellow or orange circle, causing it to become this purple hexagon. All of those steps could be called a metabolic pathway. And in the middle, you have molecules that are the product of one metabolic reaction. For example, the triangle is the product of the first metabolic reaction, but it becomes the reactant for the next reaction. And in metabolic language, we typically refer to the substrate as the we usually refer to the reactant as the substrate. We still use the word product, but if we say reactant or if we say substrate, that really means the same thing. So you always have some kind of substrate, some kind of reactant. It becomes some kind of product, but then that product is the substrate for the next reaction, and so on and so forth. So the ultimate series of reactions, we say there's one initial substrate, and the middle substrates are typically called intermediates, and then one truly end product. There are there's really three types of reactions, but two that we're going to focus on. You've seen this already. This concept you've already seen. I'll give you an example. A series of reactions, a metabolic pathway that results in the building of something, is called an anabolic pathway. And the great example of this is dehydration synthesis. So you know dehydration synthesis. I'm just going to put DS, also called condensation. That's an example of an anabolic pathway because putting together monomers to make a single polymer is a reaction that builds large molecules from smaller molecules. The opposite of that would be the catabolic pathway, a series of reactions that breaks down a large molecule into smaller ones. So an example we've already seen of that is called hydrolysis. Now, I'm not saying that dehydration synthesis and hydrolysis are the only anabolic reactions and catabolic reactions. I'm just saying that those are ones that you can immediately connect to because we've discussed those in previous chapters. But any building reaction, series of reactions that build something is anabolic. Any series of reactions that break something down is typically called catabolic. So you need to connect that vocabulary to that definition. All right. So an anabolic reaction, for example, photosynthesis, is something we will discuss in a future chapter, but it builds glucose molecules from multiple carbon dioxides. So you start with 6 carbon dioxides and you get the equivalent of 1 glucose from that. So that's a type of synthesis reaction. It's a little different than dehydration synthesis, but it's building something from something smaller. Glucose is larger than carbon dioxide. A catabolic reaction would be a breakdown. We are going to learn about this. Cellular respiration is defined as the breakdown of glucose into the carbon dioxide components. All right, so some examples there that you can start to associate. So I've given you two, really. Anabolic reaction could be any kind of dehydration synthesis, so the building of a polysaccharide, the building of a polypeptide, the building of a nucleic acid. Those would all be anabolic. Or, and now I gave you another one, photosynthesis. Catabolic reactions would be the hydrolysis of a polysaccharide, or I gave you a different one here, cellular respiration. So start to make connections between which of these are anabolic and which are catabolic. So in order to have these metabolic reactions in the cell, because metabolism would be the sum total of all the catabolic reactions and all the anabolic reactions that are happening all at the same time, really. Or the same would be true in the whole body, in the organism. But these are all, fundamentally what this is all about is the transfer and transformation of energy, which is very intangible for when you first start learning about energy, or even when you've learned about it for a long time. It feels not very tangible. Like I can build a model of a molecule, but I can't, there's no way to have a model of energy. It's, well, you can't build it and look at it, in a model kit, for example. So molecules and structures are easier sometimes because they're more tangible than the energy part of this. So energy has a lot of definitions. Sometimes energy is called the ability to do work. I never found that definition very helpful. It is a definition you will see often, but I never found it very helpful. The ability to do work, okay. But I do find it kind of helpful to kind of break energy down into forms. There's two fundamental forms of energy. One is called kinetic, and that's the energy of motion. And it can be anything moving. It can be an organism moving. It can be molecules moving. It can be anything moving, motion. Light energy is also considered kinetic. So light energy, remember energy from the sun? Light energy from the sun, that would be classified with kinetic energy. And it moves from the sun to the earth, in a sense. We'll talk about ways to think about light energy later. But right now, just put light energy into this category. So heat is when molecules are moving. Anything actually moving is kinetic energy. Light, these kinds of things are all kinetic energy. Okay, so that's one type. And then some examples. Potential energy is the other kind. It's typically defined as the energy that matter has, that something has because of its position or structure. Location, position, or structure. So it's not something that is actually moving right now, but it's like poised, like it could move. And so that's potential energy. Now, things that are a good example of potential energy would be water behind a dam. So here's the picture up here. This is a dam. I don't know what dam it is. It doesn't say. But it's like a wall, and there's water behind it. And so the water behind a dam is kind of up high, and it like, if we punched a hole in that dam, the water would start to move over the dam or through the hole. But it's not right now. So the water, it has potential energy, but it's being held still right now. But it has that position, it has the potential to move. So water behind a dam is potential energy. Another thing that's considered potential energy is covalent bonds, bond energy. So bonds, covalent bonds in a molecule, because they have electrons. Bonds or bond energy is another one that is considered potential energy. So the chemical energy within a molecule, within the bonds of a molecule. So potential energy is like water behind a dam. It could be like a rock at the top of a mountain, but it's not moving, but it's like maybe, a little bit of a breeze and it might get started. But that potential, but it's not actually moving. Now, let's say you have a rock at the top of a mountain, it has potential energy. And let's say it does fall and starts to move. The potential energy is decreasing as the rock moves down the mountain. But what is increasing is the kinetic energy because it's moving faster and faster. The problem is when it gets to the bottom, it has neither kinetic energy nor potential energy. The energy has been released, I guess you could say, from the rock out into the environment, which is, like I said, kind of weird to think about. But when it's at the top of the mountain, it has it has potential energy. When it starts falling down the mountain, it has less and less potential energy, but more and more kinetic energy. But then when it hits the bottom, it has no energy in the sense of my example. So it's odd. Energy is sometimes odd to think about. All right. I am not going to cover redox reactions, so put an X over this slide. Okay, so let's talk a little bit about free energy. Again, that's the only kind of energy we're going to talk about, really. Free energy is the energy that is able to do work. For our purposes, everything's going to be relating to free energy, so I really don't think it's important for us to distinguish types of energy in this sense. The guy who, there's an equation that goes with energy, and the guy who is credited with understanding some of this is Dr. Gibbs. I don't even know his first name. But so it's usually called Gibbs free energy. I might use Gibbs free energy, but the reason we use the letter G to represent free energy is based on Gibbs. Anyway, and here's the reaction, but you don't need to memorize the reaction. But Gibbs free energy has to do with temperature of the environment, something called entropy, which is disorder, and the temperature. All right. But don't memorize that reaction. So G is free energy, Gibbs free energy, and delta G is the change in free energy if we compare what free energy there is in a system at the beginning of an event and what the free energy is at the end of the event. All right? So some reactions, and again, we're really going to focus on chemical reactions. So let's think about metabolic reactions, chemical reactions. You know that there are reactants and there are products. And you'll remember there's an arrow. When we write a chemical reaction out, we put an arrow in the middle, and that means bonds are broken and bonds are formed. So remember I said bonds have energy? When bonds are broken, energy is released. When bonds are formed, energy is taken in. And so when you have a reaction that ultimately breaks more bonds than it forms, then more energy is going to be released ultimately than what is absorbed. So let's say you have a polysaccharide, and the polysaccharide, that's what I'm trying to write there. And you remember the glycosidic linkage holds together the monomer units. And once that polysaccharide is hydrolyzed, then all the bonds, all those glycosidic bonds are broken. And when you break bonds, energy is released. And the products would be a whole bunch of monosaccharides. So I'm just giving you an example to connect. Bunch. So one polysaccharide, and then at the end you have a bunch of monosaccharides. So when the glycosidic bonds are broken, energy is released. And it just goes like out into the universe. I mean, it's still somewhere, but it's not connected to the monosaccharides that are left. So the polysaccharides, because it had those bonds, had more energy than that pile or that bunch of monosaccharides has at the end of the reactions. And the energy that was in those glycosidic linkages has been released out into the universe, so you don't measure that. So we measure the amount of energy in the monosaccharides, and you get a certain amount. But if you had measured the amount of energy in that one polysaccharide, it would have been a lot more. So what we say is that the products have less free energy than the reactant. And that means energy has been released. And by definition, what that means is the change in free energy is negative. Delta G, change in free energy, is negative. And you guys remember, anything that's delta G means the energy of the products, I'm going to put P, minus the energy of the reactants. Anything that's a change of delta is defined this way. Like when you did slope of a line, delta x is x2 minus x1. Delta y is y2 minus y1. So it's always the second thing, the products, minus the first thing, the reactants. So the delta G, the change in energy, is a negative number. Or almost, if you draw a line here, you see how this line has a negative slope? It works too. That math helps you there. Now there is a little hump here. We're going to talk about this little hump later. But right now we're just looking at the beginning, what you start with and what you end with. We're not really going to put actual numbers to this. So you're not really going to be solving like math equations. It's all theoretical. So it's all these kind of very generalized things that you need to know. So, all right. Now some reactions, let's think of the opposite of what the example I just gave you. Let's say you had a pile of monosaccharides, a bunch of mono, or, maybe we could do amino acids. Let's change it up a little bit. That's a bunch of amino acids. I put AA. So you have a big pile of amino acids. Through metabolic pathways, the amino acids are joined together to make one polypeptide. If you measured the total energy of the polypeptide, because it has those bonds, those extra bonds between the amino acids, it has more bonds than the amino acids alone, then it has more energy than the pile of amino acids has. So somewhere energy had to have been put in, energy from the universe. Again, ultimately it's from sunlight, but we'll talk about that later. Energy came in somehow in order to create those bonds to make the polypeptide. Now on this one, if you do delta G equals, delta G, G final, or G products minus G initial, the products minus the reactants, or final minus initial, then that's going to be a positive number. And you'll notice this line, if we connect the two, you have a positive slope. Again, we're not going to worry about the hump here yet. So we say this kind of reaction has a positive delta G number, or delta G is greater than 0, positive. So any reaction that has a delta G number that is less than 0, negative, is called an exergonic reaction. All right? And these are breakdown reactions. And they release free energy. So it's almost helpful to make a chart. Exergonic reactions have a negative delta G and they release free energy. The reactions that have a positive delta G, they're called endergonic, and they have, we say they absorb energy or they require energy as a part of the process. So an exergonic reaction releases energy, an endergonic reaction absorbs or requires energy. So those are really fundamental. Now think about it. We talked about breakdown reactions, anabolic and catabolic. Anabolic is building reactions, synthesis, and catabolic is breakdown reactions. So breakdown reactions, catabolic reactions, are typically exergonic. So these are catabolic. Catabolic reactions, strictly speaking, release energy. All right. So you go from something that is ordered, constructed, to something that is just a pile of pieces, a pile of monomers. So a breakdown. Building reactions, you start with a pile of pieces and you build a structured polypeptide that's more organized. That is endergonic. That was anabolic. So since we're learning these fundamentals, at first we're going to learn them in a very black and white kind of way. Endergonic reactions require energy. They have a positive delta G. They tend to be anabolic in nature. You know, all of these things. Exergonic reactions are catabolic. They're breakdowns. They have a negative delta G. All right. Okay, so what is that hump? All right. All reactions, no matter what, and you'll notice it's on both of the previous slides, there's always a hump. All right. The hump, and we worry about the hump the most with exergonic reactions, by the way. Where's my pen? So really we worry about the hump the most here. All right. Okay. An exergonic reaction is a breakdown reaction. And in the rule of the universe, breakdown reactions are typically referred to as spontaneous. But they're not truly spontaneous. So, that, you know, thanks a lot for using that word, right? Because spontaneous makes it seem like they just happen automatically. They happen automatically once the reaction gets a little bit of energy put in. So a little bit of energy has to be put in. There has to be an absorption of energy or an input of energy, like we talked about in the endergonic reaction. And then once it gets here, then it can go spontaneously. And the equivalent to that in a real-world example would be a rock doesn't just start rolling off the top of the mountain. Somebody has to push it, or the energy of an earthquake has to push it. Something has to start it rolling. And so that initial hump, that initial input of energy to get the ball rolling, almost literally, is what we call activation energy. And activation energy is this hump that keeps reactions from happening all the time. Because the cell doesn't want every reaction to happen all the time. It's very controlled. So when there's a reaction that needs to happen, the cell can put energy into that reaction, and then it can get over that hump. It can achieve the activation energy it needs to then complete. All right. So activation energy is the distance between, it's really the distance between the amount of energy in the reactants and the top here. When you measure the amount of energy here, that's technically the amount of energy you need to put in. Then you notice you get that amount of energy released and more, because the products go even further down. So your final delta G is the net release of energy from the reactants, whereas the activation energy is the distance between the reactants energy and what's required to start the reaction, which is this amount up here. So the reactants have to absorb that much, and then when they start going through the reaction, they release that and more. All right. So the way that activation energy is controlled by the cell is that the cell will have, if it wants a reaction to happen, it will, actually what it usually does is we say it lowers it. It provides an enzyme for the reaction. And what an enzyme does functionally is lower the activation energy. And really, you can think of with an enzyme, the reaction has, just think of it being very small, almost nothing. So without the enzyme, usually the reactants by themselves aren't going to be able to get over this hump. They're not going to be able to absorb that much energy without help. But with an enzyme, the way the enzyme helps is that it lowers, we say it lowers the barrier. It lowers the activation energy barrier. Okay? So, all right, so you need enzymes to make reactions happen. You need energy transfers and transformations to make reactions happen. And the reactions themselves, you start with a reactant, sometimes we call it a substrate. It undergoes bonds breaking and bonds reforming, and then you get a product. So the arrow in the reaction really has a lot to do with the energy, and it really has a lot to do with the bonds breaking and forming, and whether or not the reaction gets across the activation energy barrier. All right, I think I'm going to take a break at this point and let you ponder some of these concepts.